A very good morning, my dear students. I hope you're well and happy and ready for this lecture. This is an important lecture again. This is the lecture on gram-positive cocci, which form a big component of bacteria which cause disease in man. So the first family we will discuss is family Micrococci, and the important bacteria here is Staphylococcus, which is a gram-positive cocci in clusters. So if you see the photo here, you can see these purple colored cocci, which we discussed in the practical as well. And these purple colored cocci are called gram-positive cocci, and the arrangement, which you can see very clearly, is in clusters. So this is a very important classification which one needs to be aware of, that you will see one, whether the bacteria is purple colored. If it is purple colored, your mind will tell you that this is a gram-positive bacteria. The shape is circular or spherical, so you know that this bacteria is a cocci. And then the arrangement of multiple bacteria together tells you that this bacteria is arranged in clusters. Can you appreciate that? So here you have two bacteria in pairs, so you can see the individual bacteria, but there are two here, some in small clusters over here, as you can see. But the classic description of Staphylococcus is in clusters, as you can see here. So you must, whenever you see a picture like this, immediately you should be able to say that this is a gram-positive cocci arranged in clusters. The other important family of bacteria which cause disease in man are the streptococci, which are again gram-positive. They are again cocci, and they are arranged in chains. All right, so you can see the distinction between staph here, staphylococcus here, and streptococcus over here, that the streptococcus is arranged primarily in chains. Of course, if you look carefully, you will also see that the size of the bacteria is slightly smaller. So the streptococci are slightly smaller. They are gram-positive, as is staphylococcus, and the arrangement is very clearly predominantly in chains. Okay, so when you see gram-positive cocci in chains, predominantly in chains, you will start thinking of streptococci, and if you see them in clusters, you know that they are staphylococci. Now the classification of staphylococci and streptococci can very often be on the basis of hemolysis. Hemolysis on a solid medium. Which medium are we talking about? Talking about blood agar, because blood agar has RBCs, and those RBCs can be lysed by some species of bacteria, and on lysing of the RBC one can see alpha hemolysis or beta hemolysis or no hemolysis, and that no hemolysis is called gamma hemolysis. Now, Enterococcus is another group of gram-positive cocci. They are slightly ovoid as against Staphylococcus and Streptococcus, which are perfect sphere. These are ovoid, and they are arranged mostly in pairs and chains. And the species which you could remember in Enterococcus are Enterococcus faecalis and Enterococcus faecium. Now coming to Staphylococcus in detail, here you can see the image also very clearly, and you can see that they are purple colored spherical bacteria which are arranged in clusters. The family is Micrococci, and in the family we have several genera. So the genera are Staphylococcus, Micrococcus, Tomatococcus, and Planococcus. And to give you an idea that what happens subsequently, so we'll take the most important genera here, which causes the genera which is associated with disease in man, that is Staphylococcus, and the species are Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus saprophyticus. So these are some of the species which are associated with disease in man. Now, what is the characteristic of these species? So, you must understand that a family has a group of characteristic traits which are common, and then they are divided into genera which will have some traits in common, and then the species, that is the species will have some traits in common, and then there are some which will distinguish them. So there will be biochemical tests which will distinguish Staph aureus from Staph epidermidis and Staph saprophyticus from the upper two. So there are always some characteristics which distinguish between different species. So we will distinguish on the basis of family. So Streptococci will be differentiated from Micrococci on the basis of certain tests, and then certain tests will differentiate aureus from epidermidis and so on and so forth. However, the common characteristics which they will share, because they belong to the same genera, are that they are facultative anaerobes and that they are catalase positive. So if you remember, we had discussed catalase in the first introductory lecture, where we had said that some bacteria are catalase producers. So what is this thing called catalase? It is an enzyme, and what is its property? It breaks down H2O2 into nascent oxygen and water. And you know that nascent oxygen is very labile. So it will immediately bind to another nascent oxygen and produce oxygen. And you will see the bubbling in the slide, which gives you tells you that this is catalase positive. Facultative anaerobe, the word facultative anaerobe tells you that this bacteria prefers to grow in the presence of oxygen. However, if oxygen is in short supply, this bacteria will still multiply in an anaerobic environment. So you will find that many of the true pathogens are very versatile. They can grow in both aerobic and anaerobic conditions, and facultative anaerobe tells you it prefers to grow in the presence of oxygen but can also survive in the absence of oxygen. So the most common species which you need to be aware of is Staphylococcus aureus, which belongs to, of course, the Micrococci family. So the same microscopic picture will be seen for Staphylococcus aureus, epidermidis, hemolyticus, all of them on gram stain will give this picture. So it will be a gram positive cocci seen in clusters. Now when we start growing it, when we get a specimen, we see it under the microscope and then we have to culture it. And the first basic media which is used is nutrient agar, and on nutrient agar you will see such colonies, and it's a very beautiful picture which you can see here, that you can see these individual colonies. I hope you can appreciate. Now these are all individual bacterial colonies, and what you see here is the golden colored pigment which is produced by this bacteria. So Staphylococcus actually means grape-like clusters, and the word aureus means that it is golden in color. And of course the word aureus has been derived from sun, S-U-N. So golden colored gram-positive cluster-producing bacteria, that is the meaning of Staphylococcus aureus, and very important for you to remember that it is producing a pigment which is golden colored. Of course, what you understand also is that it is growing on a very basic medium. Nutrient agar is a basal medium, and it is growing on this medium. That is, it is not finicky, it does not want any great enrichments, it will grow on simple medium, and of course it is, as I've told you, producing golden colored colonies. Now we grow it on blood agar as well. And on blood agar, you will see a hemolysis called beta hemolysis. So blood agar, as you know, has RBCs, so the medium is red in color, and when we inoculate the specimen on it and incubate it in the incubator for 24 hours, what do we see? We see bacteria. So both these plates have been incubated for 24 hours at 37 degrees centigrade, and on nutrient agar, since it doesn't have RBCs, you see the individual colonies and you see the golden pigment. Now on blood agar, what do you see? You see these large colonies. You can see the pigment, it is golden in color, and also you see this halo, this nice halo around the colonies. And this halo is exactly what is meant by beta hemolysis. So beta hemolysis means complete lysis of RBCs, and you can see this large colony which is golden colored with a narrow zone of complete hemolysis of RBCs, which is termed as beta hemolysis. Okay. Now the size of the colony is also described as pinhead. All right. So 2 to 3 millimeters in size, and it is a pinhead sized colony. Now, once you've seen this picture, so you've seen the gram stain, you've seen the gram positive cocaine clusters, so you know this looks like families to micrococci, clinical sample, you will see pus cells, etc. You grow it on blood agar and you see this picture, right? So in your mind's eye, you know that this should be, in all probability, staphylococcus. And since it's beta hemolytic, since it's golden colored, you know it should, you start thinking in your mind that this could be Staphylococcus aureus, all right? But by just looking at it, we cannot confirm it. So now we have to perform tests which will tell us whether our initial assumption is correct or not. So what will we do? We will do catalase tests, which I have told you, and you will see uh, the bubbling coming out, and that tells you it is catalase positive. I will try to also send you a video so that you can see how catalase looks like, a positive catalase. And now once catalase is positive, you know that it belongs to family Staphylococci. Till here, we are not sure whether it is Staphylococcus aureus or it is some other species in the genera Staphylococcus. All right, because there are other species which can also be beta hemolytic, and we want to confirm that this is really Staphylococcus aureus. So we proceed to another test which is called coagulase positive. Now this is another enzyme which is called coagulase, and it is a bound enzyme. It is bound to the cell wall of Staphylococcus aureus. And what do we do in this? We emulsify the colony. So you've seen the colony on blood agar. We'll pick it up with a sterile loop, emulsify it in saline. All right. Now to this we will add plasma. It could be rabbit plasma, it could be human plasma, and we mix it, all right, with the plasma. Now after we mix it, we rock the slide up and down for about 30 seconds, and what should we see? We should be able to appreciate this kind of picture. That is, there is clotting of the plasma, there is clumps produced. So this was the initial picture where there is a good emulsification. The bacteria is emulsified in saline, we mix it with plasma, and then we see these clumps produced. Now why have these clumps produced? They produce because the bound coagulase interacts with thrombin. All right, so fibrin, thrombin to thromboplastin, which will then convert the fibrin to fibrinogen, which is seen as these clumps, and there is clearing in the drop. All right, so the coagulase interacts with the coagulation cascade, and this is how we say that this bacteria is positive for coagulase. And if the bacteria is positive for coagulase, it is confirmed that this bacteria is Staphylococcus aureus. Now beyond that, what can we do? We can do another test, which is DNase test. So if you remember, we had discussed DNase in terms of Streptococcus. Now you know that Staphylococcus also produces DNase. So you can do catalase, which tells you the family, all right? So Staphylococcus, the family Micrococci is catalase positive. The family Streptococci, which we will be coming to shortly, is catalase negative. Now once it's catalase positive, I have to speciate. So I will do coagulase. Coagulase is positive. It looks like Staphylococcus aureus. Now sometimes this bound coagulase may not be positive, and we may need to proceed to another test, and that other test is DNase. So Staphylococcus aureus produces coagulase, produces DNase, which is not produced by the other species of the genera Staph. All right. Now we come to the other species, which is Staphylococcus epidermidis. The word epidermidis tells you something. What does it tell you? It tells you that it's a commensal on the skin. All right, so usually not a pathogen. Sometimes it can become a pathogen. And one of those reasons is when a patient is admitted in the hospital and an IV line is put on his hand, and which can remain for a week, or if it has to stay longer, it stays for longer. And this bacteria, which is sitting so innocently on the skin, can enter the bloodstream through the IV line and produce what I had mentioned in my lecture on virulence. It can produce biofilms, and those biofilms are a problem for all healthcare practitioners because it cannot be removed. It produces bacteremia, it can produce endocarditis, and so many other infections. So that is why Staphylococcus epidermidis, otherwise just a commensal, is a problem in the healthcare setting, and we need to take all precautions, all infection control precautions to prevent this bacteria from causing infection in a patient. So again, in the picture, what do you see? It looks like a gram positive. It is a gram positive. Cocaine clusters, you cannot differentiate it on the basis of grams. So we need to culture it, all right? And the best medium to culture is a solid medium. Nutrient medium, a liquid medium will not be able to tell us how many different types of colonies there are, how many different types of bacteria there are. So one big advantage of using a solid medium is that it tells us that one, we get to know individual colonies which will all have a common characteristic. So microbiology grew because we were able to grow to isolate individual bacteria and study their properties. The other thing is, in a sample, in a clinical sample, you can see whether there is one type of colony or more types of colonies. So here you can see it's a single type of colony. All right, now if I had put it in a liquid broth, I don't know how many different types of bacteria they're growing in. I will just see the turbidity. All right, I'm telling you this because one student asked me this question, and I hope that we have understood this. I will also raise this topic in the practical, so I want you to understand the very big role solid media played in advancement of bacteriology. Now on this nutrient medium, you can see the bacteria is growing. All right, now if it is growing, what does it mean? That it is again non-fastidious, it can grow on simple medium. All right, and the color is white, which you can see very clearly here. On nutrient agar it is not so clear, but epidermidis was also called albus earlier, and that means it is white in color. What also you can appreciate on blood agar is that it does not produce any hemolysis. So Staphylococcus aureus is beta hemolytic, it is golden in color, it is pinhead in size. Now epidermidis is also big in color. It is, however, white in pigment, the pigment is white, and it is not hemolytic. So it expresses gamma hemolysis. So you can see the gram-positive cocaine clusters. You can see the white colored colonies on nutrient agar. You can see gamma hemolysis on blood agar. And can you think of which biochemical test we will put here? So the first biochemical test is to differentiate between the families. So which is the first test you would like to put up? So the first test is catalase. The second test, so the first test has to be positive, belongs to family Micrococcaceae, it has to fulfill this criteria, and it should be catalase positive, okay? And then it should be coagulase negative and DNase negative because it is not Staphylococcus aureus. So in Staphylococcus epidermidis, you will see only this picture. That is, it will be emulsified, but when you add the plasma, it will not clump. All right. So the clumping will be seen only in Staphylococcus aureus. So now, why are we reading Staphylococcus? Why is there a need for us to study this bacteria in detail? The reason is this slide, which you can see that it produces so many different types of diseases. So there is skin and soft tissue diseases, abscesses, bacteremia. Bacteremia means bacteria present in bloodstream. So I'm sure you understood from your earlier lecture that bloodstream is sterile. No organism is ever present in the bloodstream. So bacteremia is a abnormal finding. It can cause endocarditis, that is infection of the heart, osteomyelitis, inflammation of the bones, pneumonia, that means infection of the lungs, septic arthritis, infection and inflammation of the joints, and the bones. And then there are certain diseases which are induced because of the toxins produced by this bacteria. And one of the most deadly disease is the toxic shock syndrome, scalded skin syndrome, and also food poisoning. What about Staphylococcus epidermidis? It causes opportunistic infections of catheters, shunts, prosthetic devices, UTI, endocarditis. And then Staphylococcus saprophyticus, which causes only urinary tract infections, and mostly in young women. Now, what are the virulence markers of Staphylococcus aureus which make it such a successful pathogen? So one is protein A, which you have already been introduced to. It's an immune evasion tool used successfully by this bacteria. Staphylokinase is again a very smart protein. It activates the human plasminogen to produce plasmin, which is a proteolytic enzyme, and it lyses the host tissue. It lyses the coagulum, which is produced by coagulase. If you remember, it has the ability to clot. And now once lysis of the coagulum happens and there is damage to the surrounding tissue, what is the net result? The net result is it allows the bacteria to spread further. All right. And so it's a very useful tool for the bacteria to become more invasive. DNAs we've again discussed in terms of streptococci, the same function holds true. This bacteria also produces pantin-valentine leukocidin, which is a leukocidin. It destroys, damages the WBCs. And so it's a very important virulence factor in an entity called community-acquired MRSA. And how does it work? It works by forming pores in the membranes of the WBCs. If you remember, I discussed toxins in the previous slide. So the toxins produced are exfoliative toxin, toxic shock syndrome, enterotoxins, and the important word for you to remember is a superantigen. What is a superantigen? Have you been told this before? I'm sure you've been introduced to it. However, I will just tell you quickly. It's a pity that I cannot see you, I cannot ask you, because I'm sure some of you definitely know the answer. So a superantigen is something which bypasses the routine method of processing of an antigen. You know that it has to be internalized, it has to be broken down, and the specific peptide is then presented through MHC complex. However, here what it does is bypasses all that and interacts with the receptors at MHC1, MHC2, and quickly activates around 30% of the neutrophils. Usually, just about 1% of the T cells are activated. Now, here what's happening is around 30% of our neutrophils are activated, and when they are activated, they get so excited, they also release a lot of cytokines. And those cytokines lead to the cytokine storm, which is dysregulated immune response, which is the basis of the horrendous syndrome which is called toxic shock syndrome. And the person can present with so many different types of symptoms. The end result is disseminated intravascular coagulation, multi-organ failure, sepsis, shock, and unfortunately may have a fatal outcome. So this gives you this pictures telling you all the various toxins. You can go and have a look at it and see it later. So now we come to the lab diagnosis, and for that we get a sample, we quickly make a gram stain, and then we will culture. Now in the gram stain, what you're seeing here very clearly are two things which are very important. One, of course, you're seeing the gram positive foci, all right, in clusters, and also you're seeing these red structures. Now these red structures are the neutrophils, okay. Can you see the hypersegmented nuclei? So these multiple segments of nuclear segments of the neutrophils tells you that this sample is from a site of infection. It has been collected properly. There are signs of inflammation. So that means that this person's sample has been collected properly, and he is definitely suffering from some infection. So the first thing which is done is microscopy. You will see gram-positive cocaine clusters along with the pus cells. And then immediately we will culture. Now what media would you like to culture this on? You know there are two, there is nutrient agar and there is blood agar. Now in a clinical setting, we always use blood agar as a primary medium. Why do we use blood agar as a primary medium? Because in a clinical sample, we do not know whether it is staph or whether it is streptococci, or maybe there's something we did not see on blood agar, on, sorry, gram stain. So blood agar is a enriched medium. It will allow staphylococcus to grow. It will also allow streptococcus to grow. It will allow other bacteria also to grow, all right, as against nutrient agar, where of course staphylococcus will grow, but streptococcus is a fastidious bacteria, it will not grow. So the medium which we will always want to use in a clinical setting is blood agar, although you know that Staph aureus is non-fastidious and we can also use nutrient agar. And the biochemical test, of course, we've discussed already, is catalase and coagulase. This is the picture on blood agar. Now I introduce you to another medium which is called a selective medium, and the medium is mannitol salt agar. All right. So now if there is suddenly an outbreak of Staphylococcus aureus in the nursery or in the burns ward or in the ICU, so in that situation, I don't want to, of course, I'll grow it on blood agar, but I want to save some time and media, and I'm looking only for Staphylococcus aureus. All right. So I can use a medium like mannitol salt agar, and what does it have? It has salt, which is medium which is making this media selective. Only Staphylococcus genera will be able to grow in a medium which contains salt. So I don't want to waste my time on other bacteria which may be present. I want to just look for Staphylococcus. So all staph, all Staphylococcus will grow on a medium containing salt. What else does it have? It has mannitol. Mannitol is a sugar. Some bacteria in this genera will ferment it, some bacteria will not. So what you can see from this picture is that mannitol has been fermented in this segment, and that tells you that Staphylococcus aureus ferments mannitol, it grows in the presence of salt. On the other hand, Staphylococcus epidermidis will grow in the presence of salt, all right, but it does not ferment mannitol. The uninoculated medium is this, so it's a pink colored medium, all right, and when mannitol is fermented, the pH comes down and we see yellow colored colonies, and those yellow colored colonies are because of the fermentation of mannitol, and the pH has come down, and that means we need an indicator which will change the color of the medium when the pH becomes acidic, and the indicator here is phenol red. All right, so this medium has three things. It has salt, which makes it selective, allows only staph species to grow. It has mannitol, which is fermented only by Staphylococcus aureus. But if I had not added a pH indicator like phenol red, I would not have been able to see the change in pH. All right, so do understand that the three components make this a very useful selective medium when I'm studying only staph, and I want to look only for Staph aureus, so I use this medium. Now the other problem, the important thing is that Staphylococcus aureus is not only a successful pathogen, but it is also developing resistance to a lot of antibiotics. And one very important criteria which we use is the ability to be resistant to oxacillin or susceptible to oxacillin. But the word here is MRSA, which means methicillin resistant Staph aureus. So methicillin is an excellent drug, and oxacillin also belongs to the same group. So methicillin was is used to distinguish whether the bacteria is susceptible to methicillin or resistant. And the antibiotic we use is actually oxacillin, but the word we use is methicillin resistant Staph aureus. So we can use oxacillin or cefoxitin to distinguish. So if it is resistant to oxacillin, we call it methicillin resistant Staphylococcus aureus. We can also use cefoxitin, but the word which we will use all the time is methicillin resistant. So we can distinguish this on the basis of phenotypic methods where we're using oxacillin or cefoxitin list, or we can simply do detection of the MECA gene, and that can be done by gene expert very rapidly within 2 hours. And beyond looking for the MECA gene, we can also look for the entire chromosomal cassette, which is called a staphylococcal chromosomal cassette, and there are 6 different variations, and these cassettes distinguish between hospital-acquired MRSA. The first four are hospital-acquired, and the last one is the community-acquired MRSA has the sixth staphylococcal chromosomal cassette. And this picture is telling you exactly what I wanted to show you. The, the phenotypic detection of MRSA. So I have used, this is a lawn culture. Lawn culture means that the bacteria is, plated to form a kind of a lawn. And once we have done the plating in the form of a lawn culture, what do? We put a disc of oxyacillin. This could be a disc of oxyacillin, it could be a disc of cefoxitin. And then we incubate it. And the next day, we open and see. So this is the oxyacillin disc on two different strains of bacteria. You can see this particular partition here. And then the next day, what do we see? We see that the bacteria is growing right up to the disc over here, right? You can see that. While on the other hand, in this particular strain, you can see that there's a nice broad zone of inhibition here, right? So this bacteria which is growing right up to the disc is actually resistant to oxyacillin, and it is termed methicillin resistant Staph aureus. On the other hand, here it is sensitive to oxyacillin, and the term used is methicillin sensitive Staphylococcus aureus. Okay. What else can we do to type? We can do phage typing, and that was a very useful tool. However, with the molecular tools coming in, this is not such an important tool anymore. However, what you're seeing here is that same lawn culture of a particular strain of staph has been done, and then 23 different phages. Phages means a virus, and which virus are we looking at here? A virus which has the ability to lyse a bacteria. All right, it kills the bacteria. So we have in each of these squares, so you have these squares, you've seen them. In each square we have plated or placed a particular strain of virus. All right, and then again we've incubated it, and the next day we see which virus killed the bacteria and which didn't. So in all these strains, let me change the color. In all these squares, what do you see? You do not see any lysis. Yeah. However, in these squares, you can see the lysis. And so that means that this particular virus has the ability to kill the bacterial strain. So the number of viruses which killed the strain is the phage type. Okay, so all these have a particular number, and then they are noted down, and it is said that this particular strain of Staph aureus is this particular phage type. Now, today's world is a very well aware world. We know that there are bacteria which are bad, and we know definitely that MRSA is a bad bug. It's a super bug. So this gentleman was actually slated for an operation. Rather, this lady was slated for an operation, but unfortunately she was found infected with MRSA, right? And the doctor said that we can't have your operation right now. We need to treat you for this bug, and then we will slate the operation. So of course she's very happy here, but there are many situations where the surgery is absolutely necessary, and one has to go ahead with it, and one tries to take all the precautions to prevent infection. Sometimes the risks are very high. The person is immunocompromised. We cannot take the risk of an operation, and we have to postpone it. All right. So MRSA has a lot of connotations for healthcare, and we need to try to prevent such infections. So what is the treatment in case of Staphylococcus aureus? So of course, you know the wonder drug penicillin, all right? And then you come across all these many drugs. Methicillin we have already been introduced to, macrolides, fluoroquinolones, aminoglycosides, vancomycin, linezolid, telavancin, daptomycin, ceftaroline. All these are drugs which are very effective in Staphylococcus aureus. However, The problem is when the bacteria is diagnosed to be MRSA, then all these drugs which I have listed on top become ineffective. And the drug to be given in such a situation is vancomycin. Okay, have you understood? If it is MRSA, then we do not give the drugs mentioned above, we give only vancomycin. We can, of course, also give these drugs, but we want to save these for the future because this fight between man and bacteria is continuous, and unfortunately, the bacteria seem to be winning. All right, and so we need to preserve our antibiotics. We need to be careful about how we use them. When will linezolid be used? Linezolid should be used only if you're thinking of vancomycin resistant. So VRSA means vancomycin resistant Staph aureus. VISA means vancomycin intermediate Staph aureus. So this was the evolution of drug resistance. Penicillin was the wonder drug. Everybody loved it. Staph aureus got killed. But then Staph aureus changed its act. It became resistant to penicillin. And then methicillin was introduced to bypass this penicillin resistant Staph aureus, and people felt, wow, we've got the wonder drug, and now we don't need to worry. Unfortunately, Staph aureus became resistant to methicillin too. So till we were at this point, what we need to understand is that penicillin, methicillin all worked where at the penicillin binding protein. It's a cell wall. So it's a bacteria. The penicillin is a bactericidal antibiotic. It works on the cell wall. All right. Now this bacteria understood what's happening, and what did it do? It initiated a chromosomal change. All right, and the chromosomal change led to a change in the cell wall of the bacteria, and so methicillin could no longer be functional. And then we had to come across this particular drug was introduced, which is called vancomycin, which could obviate the shortcomings of the penicillin group. Unfortunately, as you can see, that the bacteria is still evolving and still developing resistance, and now in 2002 vancomycin resistant Staph aureus came in. So with MRSA, one needs to take effective infection control procedures. All right. So personnel, healthcare personnel with active Staph aureus lesions and carriers have to be excluded from patient caring areas. How do we eradicate MRSA? By topical application of mupirocin to nasal and perineal areas. We need to use contact precautions, hand hygiene before and after patient contact, and wearing gloves and gowns, etc. This is just a diagram telling you about the issues with Staph aureus in a humorous way. The 5 moments of hand hygiene, where a healthcare practitioner has to follow these movements so that we do not infect an already ill patient with a superbug. These are the 7 steps of hand hygiene which you need to be aware of. Please see videos of this, and so that by the time you reach the next phase, you are well conversant with ways of protecting the patient. We come to the next group, which is Streptococcus. Streptococcaceae family has the following genera: Streptococcus, Enterococcus, Aerococcus, Pediococcus. Again, as you can see in the gram stain, it's gram-positive foci in cluster in chains. Sorry, and the species of Enterococcus, just to give you an idea, is Ephicalis ephysium. And the important biochemical for this family is that they are catalase negative. So we know that they're gram-positive foci in chains. They can be beta hemolytic, alpha hemolytic, and gamma hemolytic. So you can see that this one is beta hemolytic, right? This is alpha hemolytic, where there is partial lysis of RBC. So the zone of lysis will have green colored pigmentation around it. And here you will see, of course, no hemolysis. So the important thing here is that it is catalase negative. This classification is a good classification which distinguishes between alpha hemolytic, beta hemolytic, gamma hemolytic streptococci. So the beta hemolytic are the most important, which we're going to look at in the center. So Streptococcus pyogenes is group A, Streptococcus agalactiae is group B. The one biochemical which has been placed here is bacitracin sensitive. Then we have alpha hemolytic, where Streptococcus pneumoniae, Streptococcus viridans have been placed here. You can have a look at the biochemicals, which I'll be teaching you subsequently. And then Enterococcus doesn't produce hemolysis. So how do you distinguish between staph and strep? A quick slide. You already know staph, so I'll just see. We read out beta hemolytic. Beta hemolytic is fastidious, has pinpoint colonies as against staph, which has pinhead. It is not pigmented. It has a much broader zone of beta hemolysis than of staph. The colonies are translucent. It is easily emulsifiable. It is catalase negative. And the important thing here is that it is sensitive to penicillin. It retains sensitivity to penicillin as against Staphylococcus aureus, which you know has developed resistance to many of the penicillin group of drugs. So coming to the beta hemolytic streptococci, which are the most important species of this family. So Streptococcus pyogenes, Strep A galactiae, it is galactiae the species. It is fastidious. It produces beta hemolysis due to a toxin called streptolysin O. The biochemicals are catalase negative, bacitracin sensitive, PYRA is positive, CAMP negative. What are the infections produced by this bacteria? It could be respiratory infections, both upper and lower, skin and soft tissue infections, necrotizing fasciitis, which is a medical emergency, puerperal sepsis, bacteremia, and another important term is non-suppurative infections. So looking at the cell wall character of group A streptococci, what do you see? You see a capsule which is produced by which is composed of hyaluronic acid. All right. Then there is this group C carbohydrate, which is a very important carbohydrate in beta hemolytic strep. There is a peptidoglycan layer, and then there are certain structures coming out of the cell wall, and that is the lipoteichoic acid and an M protein, which I'm sure you all read of in the previous lecture, and a whole lot of enzymes which are produced by this bacteria. So beta hemolytic streptococci have been classified into 21 types. They were identified by Rebecca Lansfield and they called the Lansfield group. And what is the basis? The basis is the group C carbohydrate, which I have just told you about. And the first one is group A streptococci. On the basis of the M protein, Griffith typed them into 100 different M types. And M protein is a very important virulence factor. On the basis of molecular typing, 124 M types have been identified. So what is the basis of the carbohydrate C component? How did you do it and how did you go about doing it? So one, it is immunogenic. If it had not been immunogenic, we could not have been able to classify. How was it extracted? It can be extracted by acids, enzymes, formamides. It is raised, the antibodies are raised in an animal model. So this is an antigen and specific antibodies are raised in animals and then they are detected by serological assays and A to W serotypes have been identified. This plays a very useful role in rapid diagnosis from the throat swab specimen which is now available. And why is this rapid diagnosis important? Because you know in a sore throat, most of the time the sore throat is because of a virus. But if it is a sore throat because of group A strep, we want to identify it. Why? Because we want to give antibiotics. Why do we want to give antibiotics here? Because this bacteria has a potential of causing rheumatic heart disease. So I want to give the antibiotic. So what does this mean? It means I want to identify whether it's group A strep. A rapid diagnostic tool is a rapid immunoassay and then I will give the penicillin drug. I will not give it if it is not positive because most of them are viral and you know that in a viral infection antibiotics will not work and this is also called a policy of antimicrobial stewardship. What are the virulence markers of Streptococcus pyogenes? We've done this before, I'm not going into it. Group B strep is again a human pathogen It is called Streptococcus agalactiae. It can cause early or late onset septicemia. It can cause neonatal meningitis. It can cause neonatal septicemia. So bacitracin test is sensitive in group A streptococci. And here is the zone of inhibition, which you can see. So if there is a zone of inhibition, it means it is group A strep. If there is no zone of inhibition, it means it could be group B strep. This is camp test where one has plated Staph aureus in the center, and at 90 degrees to that one plates the streptococcus, the beta hemolytic streptococci we are thinking of. And then we again incubate it at 37 degrees centigrade overnight and look at the plate again the next day. So if there is this enhanced hemolysis, all right, this enhanced hemolysis tells you that this is group B strep. And if there is no enhanced hemolysis, this means it is pyogenes. PYRA's test is positive in group A, not in group B. And this pink drops tells you it's a PYRA is positive. Alpha hemolytic strep are viridin streptococci, Streptococcus pneumoniae, and enterococcus. This is how alpha hemolysis looks, so the greenish discoloration. Coming to viridin streptococci, they are mostly commensals of the oral flora, and the species are salivarius, mutans, milleri, mitis, sanguis, intermedius. Virulence factors are, the important virulence factor is the dextrans, which are synthesized from glucose and which allows them to adhere to the host surface. So it forms a biofilm, and the most important disease which is associated with them is dental caries. All right, and however, it can also cause bacteremia, brain abscess, endocarditis. So once it reaches the bloodstream, it can cause brain abscess and endocarditis, and I'm sure you've been told about the situations where bacteremia can occur. So what is the lab D of viridin streptococci? Gram stain, you will see gram-positive cocaine chains. On blood agar, you will see alpha hemolysis, and optochin will be, they will be resistant to optochin. So optochin is a disc, or it's again a disc which can be used to distinguish between bacteria. So viridins is resistant to optochin as against Streptococcus pneumoniae, which is sensitive to optochin. And then there is this bile solubility test. So viridins is resistant to lysis by bile. So you will see this turbidity which is maintained. Okay, so when you put bacteria in a broth, it becomes turbid. And after putting bile, it remained turbid. On the other hand, Streptococcus pneumoniae became clear because bile lyses Streptococcus pneumoniae. So that's a bile solubility test. Now, this is just to share with you a little humor. What do you call soldiers for hire with pus-filled abscesses on their legs? So, mercenaries is a word which was used to hire soldiers to fight for you. All right. Now, mercenaries, MRSA, Staph aureus, produces pus, and mercenaries is just a play of words on the word mercenaries. Coming to Streptococcus pneumoniae, which is a significant pathogen, and you've been already introduced to it. It causes pneumonia, it can cause meningitis, and many other diseases. So it's a gram-positive lanceolate flame-shaped diplococci. All right, so it's flame-shaped, and it's a diplococci. So flame means exactly this, that there is a flame shaped or a lanceolate shape. So that means that the uh, sides which are opposing each other, adjacent to each other, are straight, and the other end is pointed, and it is capsulated. Okay, and the capsule has, studies on capsule have told us that there are more than 90 serotypes. It is fastidious, it grows on blood agar and chocolate agar under CO2. Alright, so streptococci, all of them are fastidious. This one will prefer chocolate agar. It also prefers to be incubated in a CO2 environment. And here you can see something interesting. You can see it is mucoid, okay? So that mucoid character is because of the capsule. And the alpha hemolysis is of course a common trait of the bacteria. And then from the mucoid state, you can see it is becoming flat in the next slide. So alpha hemolytic colonies with umboniform appearance after 48 hours is what you see here. So once it becomes flat, after a period of time, you find that there is a complete depression in the center. Can you see there's a depression in the center, and that is an umboniform appearance. It is because of the enzyme amylase. And this is a typical picture of Streptococcus pneumoniae after 24 hours or 48 hours. Sorry. So it is catalase negative, it is optogen sensitive, and again it is sensitive to most antibiotics, just like Streptococcus pyogenes and A. galactea. What are the virulence factors? The virulence factors are capsule, which inhibits phagocytosis. Serotypes 3 and 7 are the most virulent. It produces IgA protease. It also produces pneumolysin, which is a membrane damaging toxin and inhibits neutrophil chemotaxis. CRP, C-reactive protein, is a non-specific acute phase protein raised in inflammatory conditions, both infective and non-infective, and it is a very useful tool to tell us what is the prognosis like and is he improving, because once the patient starts improving, CRP will come down. What are the diseases associated? Pneumonia, meningitis, otitis media, sinusitis, osteomyelitis, endocarditis, peritonitis, a whole gamut of diseases associated with Streptococcus pneumoniae. What is the lab D? You are already aware of the lab D partly through the viridins. So you can see the optogen. So one, you see the mucoid character, which is very important, tells you that capsule is present, looks like Streptococcus pneumoniae. The gram stain is giving you again a very interesting picture. You can see the diplococci and you can see this white halo, and that white halo represents the capsule again. And then optogen disc, a zone of inhibition around it, which confirms your suspicion that this is Streptococcus pneumoniae. Bile solubility again, glycis will tell you Streptococcus pneumoniae. And then enterococcus species, which is not a very not a very successful pathogen in healthy individuals, but in immunocompromised people, in people who are admitted in hospitals, it's a very important nosocomial pathogen. So it is a ovoid bacteria, mostly seen in pairs and small chains. It is non-fastidious, it is hardy. So you will find that most pathogens which are good nosocomial pathogens, they are non-fastidious and hardy. They can survive in hostile environments. It is catalase negative. It can be alpha, beta, gamma. All types of hemolysis can be manifested. It is an intestinal commensal. So it can grow in the presence of bile, which you know is present in the GI tract. It can survive extremes of pH, temperature, salinity. The problem with this bacteria is it has intrinsic resistance to certain very important antibiotics like the cephalosporins and cotrimoxazole. It also exhibits low level intrinsic resistance to penicillin and aminoglycosides. So the lab D of Enterococcus is growing it in bile esculin, and if you see this blackening, this tells you that this is bile esculin positive. It grows in the presence of bile, NaCl 6.5%. So this is telling you that this is Enterococcus, and of course PYRAs, which I told you earlier for group A, it is also positive in Enterococcus. So a quick recap, where you see the gram positive cocci, it could be gram positive cocci in clusters or in chains. So if it is in clusters, you will do catalase, which will be catalase positive, and coagulase. If it is positive, it is Staph aureus. If it is negative, it is epidermidis. Please see the chart properly. If it is catalase negative and gram positive cocci in pairs and chains, you will do bacitracin test, CAMP test. If it is beta hemolytic, you will do bacitracin and CAMP. If it is alpha, you will do optogen and bile solubility. If it is gamma, you will do bile esculin, right? And if it grows in the presence of 6.5%, it is group D. And if it is not growing in 6.5%, it is non-enterococcus, group D, all right? So that is all about the cocci. And now I'll quickly take you to some gram-positive bacilli. So diphtheria, is a childhood disease which could lead to fatality. You can see the gram-positive bacilli, and you can see some swollen ends, and that is the word. The swollen ends means coryneiform, and that is where the word comes from of Corynebacterium diphtheriae. Another staining is Albert stain, where you see the green colored bacilli with black polar ends, and that tells you it's a metachromatic granule, and they're arranged in V and L forms, which is very typical of Corynebacterium diphtheriae. It produces an exotoxin, which is a property donated by beta corynephage. Enrichment medium used is low-flow serum slope. Selective medium is potassium tellurite blood agar, where the colonies grow as black colonies. It is catalase positive. Listeria monocytogenes has the ability to cause food poisoning, also the ability to cause meningitis. It is a gram-positive bacilli, and it has a tumbling ability which helps in identifying this bacteria. Clostridium tetani, Clostridium perfringens are anaerobes. So you can see the swollen end here in the first slide, which is the spore of Clostridium tetani. While perfringens, strangely enough, does not produce many spores, so you can see it just as a gram-positive bacilli. Bacillus anthracis is an aerobic spore-bearing bacilli. You can see the spores in the center as a rail track appearance, if you see here, that you can see the gaps, and those gaps means that there are spores. You can also see these gaps here, which means again that they are spores. So this is a graph which I had told you earlier. You will quickly have a look at it, and if you have any issues with it, you can always consult me. I am always there to help you. All the best. Just another joke. When germ relationships go bad, one bacteria says to other, "You make me sick." Thank you. If you have any questions, please share them with me. Have a good day. Bye bye.